I'm a synthetic organic chemist by training, and yet here I am today standing in front of you talking about macromolecules. Now, I have to say, it felt a little awkward initially, but then I saw this, and it made me feel better. Because, let's face it, biology is just applied chemistry. But actually, chemistry is applied physics, and physics is applied maths. So I guess I can't win in that respect. But what do we mean by macromolecules? So let's start, from stating the obvious, really. Small molecules, smaller molecular weight, typically less than 700 Daltons, ideally less than 500 if you'd like to follow Chris Lipinski's rules. But small doesn't necessarily mean simple. You can see the structure of vincristine here. That could easily take somebody a couple of years to come up with a retrosynthesis for that. Of course, classically large molecules, molecular weight, either peptide or antibody-based, anything from like 1 kilodalton up to probably 200 kilodaltons. Obviously, different manufacturing process. Cell line fermentations is a key aspect in this regard. But obviously now you have a lot of new hybrid molecules. You have, for example, these stapled peptides. You have antibody drug conjugates. You've got either peptide or phospholipid drug conjugates, and you also have antisense RNA drugs. And effectively, these are the molecules of tomorrow. This is what everybody has to start working towards and synthesizing. And I think the key challenge in the workplace is actually overcoming the barrier of what a chemist does versus what a biologist does. Because I think going forward, chemists need to think like biologists and really to work jointly together to make sure that we actually have the close understanding. So a biologist would synthesize a peptide or antibody, hand it to a chemist. Chemist would have to then synthesize a suitable linker that's going to have the right liability to break apart when they need it to look at the loading, to make sure that the molecule is actually loaded with a correct ratio of drug molecules as well. And all of these are really sort of come together to the sort of joint engineering of this new hybrid. In preparation for this presentation, I did a little bit of research in the marketplace. People always say, oh, biologics is increasing, but by how much? So in certainly the last decade, FDA approval has gone up by about 10%. Biologics now account for 6 of the top 10 selling drugs. AbbVie's Humira sold $18 billion last year, I believe. Growth in biological drugs in general is estimated at about 11%, reaching about $400 million by the end of 2019. But the one metric that I thought was really interesting is the fact that the antibody drug conjugate market is going to be expected to reach $4.6 billion, and it's growing at 30%. So there's a lot of really exciting, novel, and innovative research in this area, and we need to provide the tools for your researchers to be able to leverage this. So if we're going to synthesize, for example, a biologic molecule conjugated to a small molecule, what do we need? Well, we need to draw it, so rendering. We need to have some workflow process to actually synthesize the molecule and create or produce the peptide. We need to register it. We need to have some inventory management. We need to have some sense of genealogy between these. So let's play around with this. So this is just a screenshot of E-Workbook web chemistry. So you can see in here we actually built this technology, again using MarvinJS and JChem in the background, so for structured drawing and obviously the databasing of those. And of course, once you've actually synthesized a compound, I think in the very first version of our software, we had the ability to actually push a molecule to ChemAxon's chemical registration system. Simple one-click interface, minimal configuration, and it's done. So we can synthesize a chemical, we can get it into there. What about the biologicals? So we did 2 things with biologicals. One is we actually created a plasmid sequence renderer as an extension to E-Workbook biology. You can import sort of, FASTA, GenPep, GenBank, those sorts of files to bring them in. And of course, BioEddie. So you can now go into E-Workbook and you can actually create all of the biological molecules that you need in classic format. And of course, we've integrated to Biomolecule Toolkit as a pseudo registration system, so you can actually draw whatever you need. Obviously, you have access to all of the full libraries of monomers, and you can obviously add to them much in the same way as we've just heard from Heptares. So in terms of the manufacture, actually, of a biological, this is something where IDBS have got a lot of experience. This is where we've been working together with companies for certainly probably the last decade, really to actually help them set up all of these processes around how you actually go through and create like a cell fermentation process. And there's a lot that goes into this because you've got some sort of microbial work and cell culture work, and then of course there's the in-process analytics and the bioassay, and then the formulation development and the concurrent analytical work of that. So this is actually quite a simplified representation of what actually goes on in practice. And so what we've done in ourselves is we've created a series of tools and templates. They come together called something called the BPES, or Bioprocess Execution System, which is a package of tools that we sell, which are configurable spreadsheets for workflows where you can import the data from all of your analytical work. You've got some nice instruction sheets to make sure that users are following things in the correct sequence. You've got dynamic calculations that you can apply, integrate to all of the laboratory information, and then you can do reporting. So I'm going to play a quick video here coming up, and this is our inventory tool. So with inventory, obviously in a manufacturing environment, you need to know where everything is, where it's come from. So for example, here we've actually created a buffer. That buffer could be regarded as a parent. That parent has children, so you can see all of the individual components of those. And of course, you have this sense of parent-child genealogy across those. And that's really important because if you have a problem with something, you need to go back and trace the initial source of where that problem could lie. We also have related aspects of maybe where that has been used. So if you want to know all the experiments where that particular buffer was used, you can go in here, you can track it. And again, I'm just touching on a very minor aspect of what inventory can actually do. So this first video here is us. We're actually going in and we're going to create a buffer. So I can start filling out information about the equipment, for example, that I'm going to start using. So you can see I've got a balance meter, I've got pH meters, I've got stirrers, etc. So I start filling out and maybe even scanning barcodes of those. And I can query the inventory tool, which actually tells me, sorry, the balance that I plan to use is actually out of calibration. So I need to actually then go ahead and change those. And done the second part over here, I also have a list of the components that I actually need to make this. So obviously in a manufacturing environment, we start talking about the word of GXP, control. You also see a sort of like audit by exception, or at least a reason for deviation. So if you decided not to use the balance that was recommended, use something else, at least if you log something there, auditor later on has a record of exactly how and why you choose to change those. And you can see in here, I'm just pulling back the respective chemicals for creating this buffer. And you can tell here, and again in here, there's sort of exceptions that are loaded to maybe say one component of the buffer, for example, is actually out of expired. The key advantage of obviously pulling all this information from a production system into E-Workbook is the fact that we can actually do cross-experimental searching. So things typically tend to be done in batches. You might be interested to know, how did this batch today compare to last month and the last 6 months? So we can run an execution query on the system, which gives us a report of all of these. We have hyperlinks back to those originating experiments, and again, talking about the predecessor experiments from there. And we can then also go and have a look at some of the reports and charting. So these are just different perspectives of the information and the views, again, for quality data. And I can get some sort of metrics of my productivity over time. And of course, there's different aspects or different views you can have on those data. So what have we done now? So let's catch up. We synthesized something, we registered it. We've produced a biological, we've registered it, and we've manufactured it. Now we kind of need to start stitching things together. So this is where, for example, we have integrated the BioReg, or the BMT, directly into the stoichiometry item as an alternative lookup source. So what this enables us to do is to actually go in and say, I know what the registration ID was of my peptide, I can now bring it back, and I can now start to use this in a synthetic chemistry type context. So I've got a little video over here. We're going to search for components, and I just use my reference ID that I'm finding. I can now define what it is as a reactant, and I would then go and find the peptide as a product. And then maybe I want to just search for some other chemicals or reagents to do those, so some HOBT and probably some TBTU. So you'll see when I go back to the stoichiometry item here, we can display the information in the window. We get all of the context for those. So we, again, are using the molecular masses of these peptides, so we can actually do number of moles calculation and therefore the stoichiometry of those. But we can't display the full chain of the BioEddie or Helm sequence directly in the view. But obviously in the information card, everything is there, and you can see obviously the SVG image of the peptide structure. So the genealogy, chemicals are a little bit simple. You synthesize something, maybe you make multiple batches of it, maybe some of those batches are salts, you put them into containers, and you register them. Obviously that's ignoring any of the sort of plate handling, splitting, pooling operations. But it typically tends to be quite linear. For biological, it's a little bit more complex. So as Roland alluded to earlier on, you have DNA, which has a sequence, and that sequence is then used by a restriction enzyme to then be coupled together with a different vector to create a plasmid, which is transfected into a cell line, which is then used to produce an express protein, which is then undergoes certain other steps. And again, that's a highly simplified form of what that data model actually looks like, but it can get quite challenging. So if we think about what we've seen all together now, and if we try and wrap this up together, basically, what do we need to complete the circle? So an earlier presentation today from Boehringer Ingelheim, we heard their request for MarvinJS Plus. I would like to ask for MarvinJS Plus Bio, please. So I think if somebody's drawing something, it shouldn't really make a difference whether it's chemical or biological. You should be able to represent both together. And of course, working together to create this, like, I guess, registration system that's capable of handling like these hybrid molecules. And of course, the other aspect is to take our learnings, both internal but also in collaboration with ChemAxon, to consider like what are the customer's needs regarding bioinventory, to be able to understand not only the genealogy of this, but also to visually report the genealogy in a very simple and intuitive way. And the way that we do that, we talk to our customers. So we've done this many times. We use a lot of customer-facing forums. I personally run a chemistry forum in Europe and the US on a regular basis. And we also, again, have a biomolecule forum, which is coming up in May. We also have a search forum coming up in May as well. So if you have any interest in attending these, just grab me over a beer and we can certainly have a chat about those. In my research for this, it was around the time that it was International Women's Day, and obviously there was lots of articles and press about all these fantastic women in science. And I read the story about this lady. So this is Henrietta Lacks. So she wasn't a scientist, she was actually a tobacco farmer in America, and sadly she died of ovarian cancer in 1951. But a biopsy of her cancer actually gave rise to the first immortal cell line called HeLa. And that cell line is still in use today. Over 20 tons of cells have actually been produced from her one biopsy. And effectively, unwittingly, she basically laid the foundation for a lot of the technology that we actually use in a biological sense today. So with that, I thank you for your attention, and I'm happy to take any questions.